When a microbe causes a disease in us, or a host in general, there are five steps, if you will, for the microbe to get in, cause a problem, and then potentially, depending on what type of microbe we're talking about, get out. But sometimes the microbe's not going to get out, but we'll talk about that. The first three steps are going to be all about establishing itself as far as the microbe goes. So let's jump in. The first part about a microbe causing a disease in a host is going to be a portal of entry in order to establish itself inside of the host. So most pathogens are going to have only one way that it will get into a host, one portal of entry. Although some microbes will have multiple modes of entry. For example, anthrax can infiltrate a host through the respiratory system, as well as subcutaneously, as well as, I believe, through the digestive system. However, primarily, most microbes will stick to one portal of entry, and that is how that microbe gets into the host itself. There are typically two categories that we are going to talk about as far as microbes getting into our system, or potentially kind of being a call from inside the house. So let's talk about it. So we have exogenous infections and endogenous infections. Exogenous infections are microbes that are coming from outside of our body through a portal of entry and making its way into our body, as opposed to an endogenous infection, which is an infection from within our body. Basically, our own microbiota is turning against us. So again, the call's coming from inside the house. It's already inside of us. It's just kind of turned against us. Usually, this is a case of location. Remember that E. coli near the end of part 1 of chapter 11? We were talking about how E. coli can be an opportunistic pathogen, where if it's in our intestines, it's doing just fine and we want it to be there. We have a symbiotic relationship with E. coli. However, if E. coli moves to our urethra, that can be a really big issue. So it just depends on what we're talking about there. So endogenous, it's already inside of us, causing a problem, potentially by moving locations. Exogenous infections are microbes that are outside of our body that make their way in through a portal of entry. So let's actually name some of these portals. There are roughly two that are the most important to us. The first is our skin. Now, it's less common that the portal of entry for an infection is our skin than for the other portal of entry, and you'll see why. So when it comes to skin, as long as it's unbroken, it's a relatively impenetrable barrier. It's solid, right? So unless we have a cut or some type of penetration, infections typically don't get through our skin. They can't just phase through our pores. That doesn't happen. However, you can get an infection of a hair follicle or a sweat gland, maybe underneath your nails or something. It just depends. But usually, if the portal of entry is the skin, it's because you have a puncture or an injection, a bite or a cut or a wound. And that is how the microbe is able to get past that barrier that is our skin. The second most common portal of entry is going to be mucosal membranes. And we're going to break that down into three. Gastrointestinal tract, so essentially If you consume food that is contaminated or water, maybe you are a chronic nail biter, um, something like that, uh, you have a higher likelihood of getting a microbe that is a pathogen into your system, right? Through contaminated food, water, or by putting your hands to your mouth frequently as well. We'll talk about that term fomites here in a little bit when we talk about vectors, so don't worry about that one for just the moment. The respiratory tract and the urogenital tract, those are other big ways, but between the two, I want you to focus on that second one, respiratory tract. That is the number one most common portal of entry because it is the easiest and most frequent pathway for a microbe to enter our system. And the biggest reason is we are constantly breathing in oxygen. We can't stop breathing oxygen or we die. So we're always breathing in, meaning we're always taking in microbes through the air. So let's say someone hacks up a lung right next to you in class or on the bus or I don't know, you're at a concert or in a restaurant or something, someone's coughing next to you or sneezing or something like that, and you take a nice big breath in. Those microbes coasting along on air currents just coast right into your respiratory system. That is one of the most common portals of entry because we are constantly breathing. Now, let's say that our microbes in this imaginary scenario have made it into a portal of entry. The second step is going to be attachment to the host. We call this adhesion. Now, if the microbe, the pathogen, does not become established and attach to the host, the home team is just going to push and outcompete the pathogen from the host's body. So unless the microbe has a method and very quickly adheres itself to the host, it is going to find that there is a quick exit as soon as it enters the host body. Now, there are many ways for a microbe to adhere itself to the host, and all of it is going to be dependent on the microbe itself. Depending on what we're looking at, it may be through pili or surface proteins or slimes and capsules, maybe specialized receptors, and if we're talking about horrifically helaments, it might be hooks or barbs, maybe suckers, something like that. So again, depending on what type of microbe we're looking at is going to depend on what type of adhesion mechanism it's going to use to latch on and hold on for dear life in that second step of establishing itself inside of the host, once inside. Again, I want to reiterate this term microbial antagonism. Now, our home team, our microbiota are going to absolutely fight tooth and nail to get that pathogen out of there because it's not welcome. Our microbiome is very settled into our body and takes up all of the space that it can in those non-sterile environments. So the big purpose and kind of benefit of our home team is to antagonize any pathogens that somehow make its way and adheres into our system. So wherever those pathogens latch on, our home team is going to start fighting back and antagonizing that pathogen as best as our home team can to try to loosen it up, push it out, get it out of us because it doesn't want it there. It doesn't want to compete with these pathogens. Now, let's say that our microbiome doesn't make the grade. It doesn't do good enough to kick out those pathogens. Then we turn to our immune system, our white blood cells, our phagocytes, to deal with that pathogen. So you can kind of think of phagocytes as being kind of like little Pac-Mans that will go in and engulf, consume those pathogens and try to get rid of them. So that is our next line of defense, besides our home team of microorganisms that we live with inside of us. So again, this is that third step. This is still that microbe, that pathogen becoming established. Now it needs to survive our defense system. Now that it's entered our system, it's become established in our system. Now it needs to survive our immune system. So how does it do that? There are a handful of ways and factors that lead microbes to surviving our immune system. They are called antiphagocytic factors. So something like leukocytines, which are going to be kind of like microscopic microbe weapons that microbes, pathogens, can use to fight and kill our phagocytes, which are kind of crazy. So some bacteria are able to use leukocytines that kill our phagocytes. So Streptococcus and Staphylococcus use this when they cause an infection. Kind of cool. I mean, cool for them, not for us, obviously. The second is a slime coat. This is going to be as if the microbe covers itself in butter and makes itself too slippery to be caught and engulfed for the phagocyte, which is gross to think about, I suppose, but essentially just think of it as the pathogen is too slippery for our phagocytes to catch and then engulf. Next is a capsule. So we know that capsules make pathogens particularly stronger, especially bacteria, makes them, it's like an extra protective layer. So in this case, it is that protective layer where phagocytes are still able to engulf a pathogen, but it makes it really difficult. I kind of like to think of it like we could still eat, say, like a burger from McDonald's with the wrapper on, but it makes it a lot more difficult to consume the burger itself. Like, yeah, we could eat it, but it makes it harder. So that's like the capsule. And then the fourth is that some pathogens are able to survive inside the phagocyte. Now, this one's particularly scary to think about because it's not dissimilar to kind of like a Trojan horse, where the pathogen hides inside of the phagocyte and still goes through processes and then will hide from other phagocytes in our immune system and will grow and still spread throughout the body despite having been consumed by our immune system. So kind of like a Trojan horse, it hides within our immune system itself. So Legionella and Mycobacterium are two examples of that. And remember, you don't have to know our examples. They're just a way to help you remember some of these. All right, jumping into step 4, this is where the microbe has officially established itself entirely into our system. So it's made our way in through a portal of entry. It has attached itself and is interacting with our microbiome, kind of made it past that first line of defense. It has survived our immune system as well. And now it is actually causing the disease that particular microbe causes, whatever that may be. So we are officially sick. And the way that we become sick that tissue damage or damage in general that we see is going to happen in one of three ways, potentially. So one is directly we see that damage, indirectly through a cytokine storm. So make a little note to highlight or underline this, and we'll talk about that a little bit more here in a second, but I'm going to give you a very brief what a cytokine storm is too. And that is where our immune system or immune response is so strong to the disease, those microbes themselves, that we end up harming our own healthy cells. An example of that is Ebola, where in an effort to fight Ebola, which is a virus, our body, our immune system, attacks our healthy cells in order to stop the virus, which causes immense hemorrhaging in our own body. And that is what causes an individual who is infected with Ebola to hemorrhage and bleed out of essentially every hole to death, which is unfortunate. It's because of this cytokine storm. Our immune system response is so strong, we hurt ourselves. And then the third is an epigenetic change. So this is going to be more of like a genetic change due to so much damage from the microbe itself. And we'll talk about an example of that here in just a minute. So let's look at all three of these a little bit closer. So first is going to be enzymes and toxins indirectly causing damage from the microbes. So I know that was a lot. So let's look specifically at enzymes that are going to cause direct damage once we have been infected with a microbe. So these exoenzymes is what they are called. These extracellular enzymes are going to break down barriers and essentially chew their way through our body so that the virus itself can travel, or the microbe itself can travel where it wants to go. This is horrifying to think about. Because what this is going to kind of give us an example, or an example you can think of, is when we did the coagulase test in lab, we saw clumping of that test potentially, and that was an example of an exoenzyme causing that change. So imagine that's happening on the inside, and that is a result of this direct damage that some microbes can cause. So exoenzymes. Toxins are going to be another direct damage causing item from microbes, and it's going to be all very dependent on location. In fact, we name these specific targeted action of where the toxin is occurring. So if the nervous system is being attacked, we call it a neurotoxin. If it's the intestines, it's enterotoxins and hemotoxins, if it's red blood cells, and so on and so forth. So there are two categories of toxins, exotoxins and endotoxins. But more than anything, I just want to get across here that we are looking at the location of where that toxin is occurring. You don't have to memorize neurotoxin, enterotoxin, hemotoxin, so on and so forth. Just know that when we name a specific toxin, we're saying that that substance, that poisonous substance, is being produced by the microbe, and it's attacking a specific, usually a system of some kind, one of our systems. So let's get back to those exotoxins and endotoxins. Exotoxins are going to be made by the microbe and then spit out to be used, and will cause a variety of symptoms. Now, there's two important things here. First is that these exotoxins cause kind of a bevy or a multitude, a variety of symptoms within the host. So it's not all just one, fever and chills and body aches. No, it's going to be a variety of different symptoms based on the exotoxins. It's not all just one thing. The other thing that is important to note with exotoxins is that they are extremely powerful symptoms. So not only are they a variety of symptoms, they're going to be really intense. And that is a very big difference between exotoxins and endotoxins. Endotoxins should be ringing a bell in your head, actually. They should sound very familiar. This is going to be a product from gram-negative bacteria cell walls. So we should remember ever so slightly that the outside of the cell wall for gram-negative bacteria is going to be composed of lipid A. And that lipid A is specifically going to have that endotoxin. And remember when we talked about endotoxins before a couple chapters ago, that when the cell dies, if it is a gram-negative bacteria, this can be a big issue. So if your patient is not stable and you treat them and they have that gram-negative bacteria and you treat the bacteria and the bacteria dies and it releases these endotoxins as kind of a final screw you on its kind of like last breath, so to speak, then that endotoxin is going to be released. In that last final act of that dying bacteria, that will potentially tip your patient over into not being okay if they're already unstable, and it could potentially kill them because these endotoxins being released will cause a reaction within your patient. So let's talk about that a little bit more. So endotoxins are going to have a very select amount of symptoms, which is fever, chills, weakness, aches. If you have pregnant individuals, potentially a miscarriage or hemorrhage, system shock, and if these endotoxins build enough in this individual's system, it can definitely even cause death. So these are usually a weak toxin that have weak symptoms and a weak kind of magnitude of the symptoms themselves. But that is not to say that it wouldn't be enough to tip your patient over into dying, because enough buildup of an endotoxin can lead to a bad outcome. So remember, we have exotoxins and endotoxins, and we also have those exoenzymes as well. These are those directly causing damage items from a microbe that cause diseases. All right, moving on to indirectly causing damage. So these pathogens are not directly causing the damage themselves. They are indirectly causing damage. And a lot of this is going to have to do with our body's response to the microbe. So remember that cytokine storm. Sometimes this is in the form of a fever. It could be swelling. And again, I'm going to go back to that cytokine storm. It is our body's immune response to the microbe itself, basically causing damage to ourselves in a response to the microbe. So again, think about Ebola. Um, it is not the virus itself doing the damage. It is our body's response to the virus causing damage to our own cells. So kind of an interesting aspect of how a microbe can do so much damage, but again, it is indirectly. So that third way that microbes cause damage to their host is one we don't fully understand yet, and that is epigenetic changes in a host cell. So this is where the microbe causes enough damage that it changes the DNA of our cells, the host cells. Um, and in that instance, our cells will keep replicating with that damaged DNA. So we kind of perpetuate whatever it is that that microbe has changed within our own DNA. So this is one of the reasons why we could potentially still see symptoms well after a microbe has left the host's system. An example of this is like long COVID. So well after an individual has recovered from having COVID, we don't see that person testing positive for having COVID anymore. Um, that person is still short of breath, still having issues with their lung capacity. Um, and what we're thinking it might be tied to is this epigenetic change due to the damage that COVID made while that person was infected. So their cells just, yes, the microbe's gone, but their cells have not genetically recovered and they potentially never will. So this one's kind of a scary one to think about. And again, we don't fully understand um, this one as much as maybe we wish we did. So up until now, we were under the impression that microbes fall into one of two pathogen-based categories as true or opportunistic pathogens, since they're one or the other. However, we're kind of realizing that maybe the lines might be a little bit more blurred as far as those distinctions go, because it's not as clear cut as it could be, because there's so many factors about virulence, as well as our immune system response. So essentially what we're getting at here when it comes to pathogens that cause disease or whether disease will occur, it's all a lot more complicated than we previously thought. Before we get into that fifth step of a microbe leaving our system, potentially, sometimes they don't, let's talk about the manifestation of diseases. So what's happening at the host level, and then we'll come back to that step 5. So we're going to start looking at patterns of infection. The first part of a pattern, as far as infections go, is the where. So let's talk about local infections. This is going to be one area where the infection is limited to just that one area versus a systemic infection where the infection has now moved to the whole system throughout the body. This is usually where the infection piggybacks into the bloodstream and rides essentially that highway throughout the whole body. And then we have a focal infection. This is where a systemic infection begins as a local infection. And honestly, this is where most infections end up, where they are a local infection and they become a systemic infection. So a focal infection, but they're called something specific. The next part of the pattern is the health status of the patient prior to infection. So a primary infection is where a patient was healthy and then now they're not. Now they have this infection and they are unhealthy. A secondary infection is usually where an opportunistic infection occurs within a patient who is already compromised. So again, kind of think of like someone with HIV, their immune system is already compromised and a secondary infection comes in and essentially makes things worse. From there, we need to talk about how long and potentially how severe the symptoms of an infection are. So we have an acute disease, meaning that the infection is quick, you get sick, and you get over it. A good example, the common cold. Usually the symptoms come along pretty quickly, and then you very quickly get over a cold. A chronic disease and a latent disease both will have a very long period of time where you are sick, sometimes forever. Just depends on what we're talking about, like what disease we're talking about. With a chronic disease, however, the entire length of the infection, you have symptoms, even if the symptoms are particularly weak. And then eventually, potentially, you get over it, like typhoid. A latent disease is going to be a disease where you have symptoms for a period of time and then they go away and then they come back and then the symptoms go away and then they come back. A good example of a latent disease is herpes. Now, when an individual has contracted a disease, we can show symptoms, signs, and also we need to look at syndromes. Now, people use the term symptom and sign synonymously. However, they are not the same thing. And I do want you to know the difference. A symptom is felt by a patient. This is going to be something like pain or achiness, feeling weak, right? So this is something that is felt, whereas a sign is something that can be measured or seen by a practitioner or someone who is external. Now, a sign could be like swelling or possibly the measuring of a temperature. However, something like a fever actually spans both a symptom and a sign because you can feel that you have a fever, but it's also something that can be measured. A syndrome, however, is a group of signs and symptoms that will be specific to a disease. For example, with COVID, there was a list of things to look out for, having a fever, shortness of breath, a lack of taste or smell, as well as headaches and a handful of other things to be looking out for. Those were syndromes of that specific disease. Here are some examples of some symptoms, maybe pain, soreness, aches. Remember, these are things that we feel. Some signs, things that we can measure, an edema, granulomas, lymphodentist, just depends. Stuff you can measure as a practitioner of healthcare. You don't have to memorize these. These are just some examples of signs and symptoms. Some more examples of signs, and specifically of like a blood infection. And just to kind of emphasize that a symptom is something that you feel. You can't feel that your white blood cell levels are off. However, that is something that we can see as a sign of a blood infection. So leukocytosis or septicemia or bacteremia or viremia. We can measure the amount of white blood cells or the amount of bacteria or viruses present in the blood. Remember, signs we measure, symptoms we feel. Now, here's where things can get a little bit fuzzy, a little bit of an issue, is individuals who have asymptomatic infections. Remember when we put a in front of a word, that means no, none, or not. So we're saying that we don't have any symptoms. This can be a big problem because even if you're not showing symptoms, that doesn't mean you're not contagious if you have an infection that is contagious. For example, COVID. This was a really big issue with COVID because even if you didn't show signs or symptoms that you were sick, people were still walking around having COVID and spreading it. This is also an issue when it comes to something like measles. And one of the biggest reasons why measles spreads so easily. You can't tell you're sick yet until you are sick. So again, asymptomatic infections are infections where there are no noticeable symptoms being produced, and you can still spread it. So, those were all of those lovely ways that a microbe is going to manifest and display, if you will, sickness in a host. That leads us to that step 5, potentially, of vacating the host, that portal of exit, which typically is going to be the same way that the microbe found its way in, will also be the exit out of our body. However, not always, but usually. In communicable diseases, the microbe needs an exit in order to spread. That is what makes it communicable. It is going to enter our body, reproduce, cause havoc, and then it needs an exit so that it can find other hosts to infect. In non-communicable diseases, however, this step doesn't really matter because it's not trying to exit our body because it can't. We are a dead-end host and the last place that microbe's going to be. And then one of two things is going to happen. Either when the microbe dies, it's going to take us with it, or we survive, the microbe still dies. All right, with that, we'll put a pause in it. And that is the end of Chapter 11, Part 2. Stay tuned for Part 3. And again, this is such a hefty chapter. I'm so sorry it's so long, but that is why I'm breaking up into 3 parts. So jump in for Part 3, and that will be the final part of Chapter 11.